Welcome back to the Classics of Immunology Journal Club, a la Kendall. And this week we're in 1981 with the paper, one of my papers, again, because this is when we really started to rock and roll. This paper is, the authors are Richard Robb, Alan Monk, and Kendall Smith. And the title of this paper is T-cell growth factor receptors, quantitation, specificity, and biological relevance. And so before we get started, don't forget to, if you like this video and you like the Journal Club, click on the YouTube channel, click on the video that you like the video, click on the bell to get notified for more, then scroll down to all of the links, my own links to my website and so forth, and social media and that sort of thing. And on the website, if you click on the writing tab, you'll see all of the papers, the classic papers in immunology, starting in 1798 with Edward Jenner and the smallpox vaccine, up until, well, mostly the present time. So this paper is really, and from my lab, one of the very classic papers. Now, the background on the people involved in this whole thing: Richard Robb was a new postdoc who was well trained in biochemistry, membrane biochemistry, and he was critical to the success of this whole effort, because what we needed to do, as I'll get into, is we needed to purify our own radio-labeled T-cell growth factors so that we could generate and create a receptor assay. Now, the other person that was critical to this a whole series of experiments was Alan Monk. And I think I went into a little bit about him before, but just to reiterate things, Alan was a professor at Dartmouth, and he'd been there since the late 60s. And he'd been trained as a chemical engineer, and then he took a postdoc at MIT in a lab that was interested in hormones, which is a very unusual thing to do for an engineer. I never did ask him why he did that, but anyway, he did that. He became very interested in hormones. And when he came to Dartmouth as a young investigator, he devised the very first assay for glucocorticoid hormone receptors. And he did this assay using thymocytes as his target cells to see if he could demonstrate glucocorticoid receptors. Now, glucocorticoids are the so-called corticosteroids, or adrenal corticosteroid hormones produced by the adrenal gland, which sits on top of the kidneys. That's why it's adrenal. It turns out that glucocorticoid hormones are essential for life, and consequently, there are receptors for these hormones in every tissue in the body. And if you lack the glucocorticoid hormones, that's you end up with a syndrome that's called Addison's disease. And I think the most famous person who had Addison's disease was President Kennedy. And he was just lucky enough that he came into being and developed this affliction after the biologists had discovered ACTH, which is the adrenal corticotropic hormone that stimulates the adrenal gland to make glucocorticoid hormones. And there is a back and forth between ACTH and the glucocorticoid hormones. So Alan was well versed in the whole receptorology kinds of things, which was a unique kind of thing at that time. There was only a few other hormones that had been looked at to see if they had receptors. One of them was insulin, and another one was epidermal growth factor. And then there was a series of steroid hormones, of which glucocorticoid hormones were one. And then there was the sex hormones, testosterone, estrogen, progestin. In the introduction in this paper, we lay the whole thing out, saying that all of the background information that we had accumulated over the last 3 or 4 years in my lab at Dartmouth, that it looked as though what we were dealing with when we were dealing with T-cell growth factor activity was that this activity was most probably due to a hormone-like molecule. And the evidence for that, or the data substantiating that kind of thought pattern, was that but this time we had separated T-cell proliferation, T-cell activation into 2 different steps. One caused by the antigen interacting with the T-cell antigen receptor, and the other one, an additional one, was T-cell growth factor. Together, the antigen receptor and the T-cell growth factor would result in T-cell proliferation if you stimulated your cells with the proper antigen. And the antigen, there were two different independent events of antigen that we had discerned by that time. And one was the production of the T cell growth factor that was antigen dependent. And the other one was the acquisition of responsiveness to the T cell growth factor. Therefore, we said, well, this is what we're dealing with here is that the antigenic stimulation, which comes from an external environmental molecule or molecules, is essentially activating an internal hormonal regulatory system that actually mediates the whole immune response. Now, that was basically an anathema for the immunologists, because the immunological community had been steeped in the dogma that for years and years and years, the only thing required for the activation of the immune response was the antigen. Now, of course, that's true, but there is a molecular mechanism that involves, in this particular instance, T cell growth factor that carries out this antigenic stimulation. And so therefore, after becoming educated by Alan and also with me, and I had previously been involved or interested in this whole area when I had been in France 5 or 6 years earlier, and I had developed an erythropoietin assay for the red cell stimulatory hormone, erythropoietin. And at that time, I spent a lot of time in the looking in the literature for, what were the characteristics of hormones. And so this was sort of like deja vu all over again for me, because I'd already been over this ground with erythropoietin. It seemed to us, and it seemed to me, that what we were dealing with was a polypeptide hormone, in contrast to glucocorticoids, which were steroid hormones. And the steroid hormones were small molecules of molecular weights of around 350 daltons. Well, insulin was the only other really well-characterized peptide hormone or protein hormone, and it had a molecular weight of several thousand daltons. And so it looked like we were probably more like in the insulin category than in the steroid hormone category. We knew that the mitogenic effect, based on our Tcgf assay that we had produced, that the proliferative effect of the T cell growth factor was T cell growth factor concentration dependent. The more you had, the more proliferation you got, up until you reached a plateau and you couldn't get any more, no matter how much you added. We knew that when we tried to look at other factors and hormones and so forth, that only Tcgf would cause the proliferation of the T cells. And the effect of the Tcgf, when we had it in our culture vessel, was only Tcgf responsive cells would respond. If we used other kinds of cells like fibroblasts or whatever else, we didn't get a proliferative response. And so those were all characteristics of specific hormones. And so that's what set the stage for this particular paper. The other thing we had already done some absorption experiments in much the same way when you're looking at antibody activity and you have the source of the antigen. Let's say you have an anti-sheep red blood cell antibody, and then you have sheep red blood cells. You can mix the two together or wait for a while, and the antibody activity will bind to the sheep red blood cells. And so then you could take it off and see the disappearance, the absorption of the antibody. Well, we did the same kinds of experiments with Tcgf, and I'd already done the same kinds of experiments back in the days when I was looking at erythropoietin. And we found that only antigen-activated T cells would absorb the growth factor activity, and that the absorption was time, temperature, and cell concentration dependent, and that you could even use glutaraldehyde fixed cells to absorb the activity, which meant that we were probably dealing with a receptor on the cell surface. So it was clear, if we were going to go further in this whole story and really nail the coffin down, that what these lymphokine, cytokine activities were really hormones. They were hormones that the immune system generated and utilized to regulate itself. And so in order to go further, we needed to develop a radiolabeled hormone binding assay to bind onto our cells so that we could study the characteristics of the receptor. of the binding site. That reminds me to say that there's a difference between a binding site on the one hand, where you can demonstrate binding, but a true receptor also transmits a signal through to the cell and induces a characteristic response. So that's a true receptor. So, and the materials and methods of this paper, the cell cultures, just to reiterate, the cytotoxic T lymphocyte line, CTLL, and we also had a helper T cell line that we got from Max Schreier, my friend at the Basel Institute. We cultured those cells at a low density between 10 to the 4th per ml, 10,000 cells per ml, and 100,000 cells per ml. And for these series of experiments, but this time we had partially purified IL-2 through a gel filtration column that separates cells on the basis of size. And we also could also quantify the Tcgf in our Tcgf assay, bioassay. And so we always used 1 unit per ml. And that was our definition. That meant that the 50% point occurred at a dilution of 1 to 10. When we wanted to make human Tcgf responsive cells, we used PHA to stimulate human peripheral blood cells, and we cultured them with 1 microgram per ml of PHA for 3 days, and then if necessary, we would purify the blasts over a Percol density gradient. We also did murine Con-A stimulated cells, as well as LPS stimulated cells that was specific for B lymphocytes to undergo a proliferative response. The Tcgf assay, we used a clone of our CTLL, clone 15H, which was a tumor-specific cytotoxic T cell. And in order to make and purify radiolabeled Tcgf, we made the Tcgf from a leukemic T cell line, JERKIT, which had been established several years previously up in Seattle, Washington. And we had cloned this cell line, so we picked out the most stimulatory or stimulated clones of JERKIT cell line that would make the most T cell growth factor. We cultured the cells at 5 million cells per ml with 1.5 micrograms of THA per ml. And we cultured those cells in the presence of just 1 mg per ml of BSA rather than serum, so that we didn't have any other contaminating other proteins in there. We labeled the cells initially with S35 methionine, and we did experiments so that we would get methionine-free medium, and then we would put in our S35 labeled methionine. And it turns out that we needed to add also unlabeled methionine up to a certain concentration in order to get any Tcgf made. We also used subsequently, after we did the initial experiments of S35 methionine, we switched to tritiated leucine and lysine because there was only one methionine molecule atom in the molecule, or molecule in the molecule. And there were lots of leucines and lysines. And in addition, S35 has a relatively short half-life, whereas tritium has got, I think, a half-life of 5,000 years. So that was the way we would culture the JERKIT cells in the presence of S35 methionine or leucine and lysine overnight, 14 to 18 hours, that we would then harvest the supernatant and spin out the cells and concentrate the medium using an Amicon filtration device so that we could get this volume small enough that then we could load on to a Cepidex G100 molecular sieve column. And the yield from that column was about 75%, which is pretty good. That's not bad. We only lost 25%. We would then take the active fractions from that column. We had to assay them all on the Tcgf assay. And we would then further purify the radiolabeled Tcgf over our isoelectric focusing tube gels, and we could run the sample out onto a pH gradient, and then we could slice up the tube gel into 2 millimeter slices. Then after that, and after then identifying, then we could elute then from those 2 millimeter slices of the tube gel the Tcgf and the radioactivity. We could assay for both of those in our assays. And then we could also take the active fractions from the IEF column and analyze it by SDS page, which also then would be a molecular check on how many radiolabeled molecules do we really have, and what we hoped to be just one. We could also test that radiolabeled fractions, the fractions that had the radiolabel for other factor activities. We tested it for interferon, for macrophage specific CSF, for B cell growth factor activity, and they were negative. So that was good. And then in order to do the Tcgf binding assay, if we could satisfy ourselves that we had purified radiolabeled T cell growth factor with no other proteins there, and I'll show you the data for that, we devised a Tcgf binding assay under the tutelage of Alan Monk, who was invaluable. And we took our cells, and the binding assay was done the same way that Alan did his glucocorticoid assays in 1.5 ml Eppendorf, plastic Eppendorf tubes. We would have the cells per tube at either 500,000 all the way up to 2 million cells per tube. And they would be in a final volume of 200 microliters with tissue culture medium and 1 mg per ml of BSA. We would incubate them at 37 degrees for 20 to 40 minutes. And then we would stop the binding reaction by adding 1 ml of ice cold medium to the tubes. And then we would spin those in an Eppendorf microfuge at 9,000 times G for only 10 seconds. We would then take the soup off, save that, because that was going to be counted for the free concentration of the T cell growth factor. And then we would resuspend the cells in 100 microliters of medium, and then spin them again over 400 microliter tubes, plastic tubes, which are tall, skinny tubes. And with the mixture of silicone oil, 84%, and paraffin oil, 16%. And that had been worked out in assays that Jerry Crabtree had been doing in Alan's lab to assay FC receptor binding. And it was a good way to separate any free hormone concentration from the bound concentration very rapidly. We'd spin those then at don't have that written down. Anyway, we'd spin them fast in the microfiche tubes. We'd cut the tips of those tubes with a razor blade and put the tips into counting tubes along with scintillation fluid, and that would be the assay for the bound. So there's only two data sets, two kinds of data sets, but at each cell, at each hormone concentration, bound and free. We could also do competition assays, which we did with a whole bunch of different kinds of hormones and factors. And I'll get into that when we get into the results. We did association rate experiments as well as dissociation rate experiments at both 4 degrees and also at 37 degrees. And in order to do these experiments, we would use saturating concentrations of labeled Tcgf at around 250 picomolar with and without unlabeled concentrations of Tcgf at 25 nanomolar. And we could calculate the molar concentration because we had purified IL-2 or Tcgf and so forth, as I'll get into. We analyzed the binding. We could stop the reaction after different time intervals. And we could also, and then we'd get association rate curves. And then we could also do dissociation rates by taking a near saturating concentration of the radiolabeled Tcgf and bind it on at 4 degrees for 45 minutes. And then we could wash and resuspend the cells with and without 25 nanomolar saturating concentration of unlabeled Tcgf to promote exchange. And we could then at various time intervals find out how much was bound and how much was free and so forth. And in addition, we could take both the cells as well as the supernatant, the free, and we could see whether or not the radiolabel had been bound to protein by assaying for trichloroacetic acid precipitation abilities. And that would give us an idea whether or not the radiolabeled was being degraded in the cells or and then excreted into the supernatant. So then then we also wanted to make sure, or we wanted to find out whether or not the concentrations of unlabeled Tcgf, which would cause the cells to proliferate, was the same or different from the concentrations of the radiolabeled Tcgf that bound to the cells. So in the results, and now I want to do some screen sharing to show you these results. Okay, so this is figure 1 from this paper, which shows you how we purified the radiolabeled Tcgf. In the left, in the A portion of this figure, we've got the Tcgf activity in dashes. And so this is the activity, and you can see it's concentrated in just a few slices, elutions from this column. We used these proteins up here to standardize the molecular size of each of the fractions coming off of this column. And we could also then count each fraction to see what the radiolabeled S35 methionine, and you can see that the peak here coincides with a peak of CPMs here, not a small peak. And so then we took these fractions and then loaded them onto tube gels for isoelectric focusing, and with a pH gradient here between 6.5 and 9. And so you can see again, the CPMs of the radiolabel are in the solid lines. And so there's a big peak right here of radioactivity. And the Tcgf activity is in the dashed line here. And you can see it exactly coincides here. There's also a peak of radioactivity and a peak of Tcgf activity right here, but then the two separate after that. So we would slice these gels into 2 millimeter slices and then elute off in order to get this plot here. Elute the protein from each of those slices, and that's then what we took and examined through SDS polyacrylamide gel electrophoresis in the figure 2. And so these are our protein standards over here, reduced with 2-mercaptoethanol. And then this is the S35 methionine unreduced. It comes out at 14.8 molecular size and 14.8 thousand molecular size. And then with the reduction, and then therefore the breaking of the internal disulfide bonds, we lengthen out the whole molecule into 15.5 kilodaltons. So that's what we used to bind onto the cells, and we were very pleased with that. So this is an experiment whereby we looked at the time course of the association and the dissociation. And in the top panel, you can see that is the association rate of the radiolabeled T cell growth factor at 37 degrees and at 4 degrees. And so when you cool the system down, it takes a little bit longer to come to steady state. This happens within 15 minutes, and this takes about an hour to come to steady state. And then here is the dissociation rate that we did in this particular, these kinds of experiments, where we bound on at 4 degrees for an hour, and then we would add an excess of unlabeled Tcgf to promote exchange and promote the elution of this whole thing after we diluted it out. And so this is at 4 degrees, and this is at 37 degrees, and this is Tca precipitable right here. So you can see that the difference between here and here is that there's a lot of radiolabel that is not associated with protein anymore, still in the cells, but it's not associated with protein anymore. And that was just the beginning of a whole series of experiments to pin down exactly what was going on in there. But basically the bottom line is that the Tcgf was binding very rapidly, and it was coming off very slowly. So that's how you create a high affinity receptor, essentially. The next thing we wanted to do is to test different cell lines and cells for radiolabeled Tcgf binding. And in the top panel here, panel A, we've got PHA blasts, human PHA blasts. And then we've got, make sure I don't give you the wrong data here. We've got the helper T cell line here, and here are the CTLLs. And you can see that you can have, this is the amount of bound IL or Tcgf. And as it reaches steady state, you can extrapolate back and get a reasonable estimate of the number of receptor sites per cell. And this is the free concentration down here on the x-axis. Or you can take the bound and make a ratio of bound and free at each concentration and plot that against the bound molecules. And this is called the method of Scatcherd that was first published in 1949, just to give you an idea how long these hormone people have been dealing with these issues. And you can see what happens with the data then. If you're only dealing with a single affinity of a receptor, you get a very straight line that proceeds down and passes or bisects the abscissa, and then you get a direct and very accurate readout of the number of sites per cell of the binding sites. And so this is the PHA blast, and this is higher than the helper T cell line, and which is higher than the CTLL cells. And so then we went on from that after we'd established those kinds of characteristics, and we looked for Tcgf binding to either unstimulated or stimulated cells, knowing that when we added partially purified or purified Tcgf to unstimulated cells, we did not get a proliferative response. So the prediction would be is that we shouldn't see any binding sites on those, and that's exactly what happened. Here are mouse cells, splenocytes and thymocytes, and the number of sites per cell were undetectable with a detectable level of 60 sites per cell. And then if we activated those cells, so either CONA or alloantigen and MLC, that we would get very readily detectable 50 to 100-fold increase in the number of detectable binding sites per cell. And in this particular situation, it looked like the equilibrium dissociation constant, or the affinity constant, was around 20 picomolar, 20 times 10 to the minus 12th molar. If we activated the splenocytes with LPS, then again, we found no detectable receptor sites per cell. However, we didn't in these series of experiments, we did not activate the the splenic B cells with other mitogens, such as anti-immunoglobulin, activating specifically the receptor for antigen. And we should have, because we would have found later on, we found Tcgf binding sites on B cells. Then if we look at CTLL2 clone 15H, we could find easily detectable sites. These were glutaraldehyde fixed cells. They were positive, just the way they had absorbed. The helper T cell line was very positive. Human cells, either unstimulated, we had detectable sites per cell, 200 in this particular experiment, with a equilibrium dissociation constant of 7.4. And that's by comparison to when we look at PHA stimulated cells or alloantigen stimulated cells, then we had 5 to 10,000 sites per cell. And we had readily calculable equilibrium dissociation constant of around 5 picomolar. So we didn't know whether or not this could have represented, a very low number of binding sites, say 200, on all the cells, or it could mean that there was a very small fraction of cells that had, say, 10,000 binding sites per cell or more, and we couldn't distinguish at that particular point in time. And that's a very important point, as we'll find out as we go along in these kinds of experiments. So then we went on to look at some malignant cell lines. And I want to go on to table 2 here, as we looked at a whole bunch of T cell lines. All of these are, and they're all basically human T cell lines from leukemic patients. And they all had undetectable sites per cell. There was only one out of this series, HUT102B, that had readily detectable binding sites per cell with a KD that was just like the normal cells. We looked at 4 B cell lines that were common B cell lines. Everybody had them in their lab, undetectable. And then we looked at a myeloid cell line, a promyelocytic cell line, a monocytoid myelomonocytic leukemia. They were all negative. There was one cell line that had come from a gibbon ape leukemia called MLA144, and that had readily detectable binding sites. Turns out that the KD, or the dissociation constant, was a little bit higher here, but it wasn't, we didn't really remark on that. And if you go back to see the scatter plots of HUT and MLA, you can see the difference. I think this was HUT, this was MLA, and in the saturation binding curve, and then the scattered plot, the steepness of the slope, the higher the affinity. So these are the human. This is the HUT102, and this is the MLA144. And you can see that this has a little bit lower affinity, not markedly, and we didn't know what to make of that because this was from an ape and so forth and so on. So we then went on to say, okay, let's see if we can establish hormone specificity for these binding assays. And so we looked at a whole bunch of different hormones using either mouse cells or human PHA blast cells, depending on the source of the hormone. And so we looked at, on the mouse, EGF, NGF, MSA, multiplication stimulating activity, fibroblast growth factor, LAF, lymphocyte activating factor, that we now know is called interleukin-1, and interferon alpha, interferon gamma, and so forth. We also then looked at erythropoietin and using human PHA blast cells and insulin, and they were negative for percent inhibition. We looked at Jurkat Tcgf, and of course, the Jurkat Tcgf would compete. And we also looked at since Jerkett was a leukemic cell line, we wanted to look at also TCGF coming from normal human cells. And from the isoelectric focusing, we knew that there were 3 different peaks at pHs of PIs of 8.2, 7.9, and 7.6, and they all equally competed for the growth factor binding. So that was interesting to us, and it's because it established the hormone specificity of the binding. Now, and so then we went on and this just shows the species specificity of competition for binding. And so what we've got here are PHA blasts with unlabeled TCGF activity added into the binding assay. And as you add in more and more unlabeled TCGF, you compete away the maximum binding. And so then this is the same thing of human TCGF onto CTLL cells in the open circles here. And then this is rat TCGF binding onto human PHA blasts and also onto murine CTLL2 cells. And so rat will bind to mouse, but rat will not bind to human, and they don't cross-compete. So that was interesting. And in the hormone literature, there's always, very often there was noted that they did not cross species barriers. Then the final things are, the major question then to establish this whole assay system was, the question was is the concentrations that lead to the biological proliferative response using unlabeled TCGF similar or different from the amount of binding to the receptor? And you can see in this particular experiment, they were quite similar, with only a little bit of variation at the 50% point between the binding curve and the biological response curve. And we went on to repeat this many, many times, and they basically were overlapping and coincided with one another. So I'm going to stop screen sharing at this point in time and come back to you. So that was it from the standpoint of the results. I remember when I was writing on this paper, it was in July, and I always like to be outside when I wrote in the summertime, because New Hampshire's a pretty cold place to live. And so when the sun finally did shine, just briefly, I would be outside writing, either in my garden or any other place I could find to work. And so I remember working on this paper and saying to myself, well, this is just too good to be true almost. I mean, this is just fabulous kinds of data. In the discussion of this paper that I was working on in the garden, basically what we established several different points. One is that all of the data that we were able to generate were indicated that the binding site detected is on a receptor that initiates T cell proliferation. That was very important. And the lack of detectable binding to mouse cells and human normal unstimulated cells, unstimulated mouse, unstimulated humans, was consistent with the lack of the proliferative effect that we found when we would add TCGF to the unstimulated lymphocytes. The caveat there, as I've already mentioned, is that there were about 200 sites per cell on the human unstimulated PBMCs that we used in these assays. And that turns out to be very important as time goes forward. Thirdly, in this discussion, when you activated the T cell receptor with antigen, it really markedly increased the number of detectable receptor sites on these cells, about 50 to 200 fold. The lack of these detectable sites on the LPS blasts, I was very happy about that, because we'd already named this factor the T cell growth factor. So I didn't want or didn't expect there to be TCGF receptors on B cells, and that's what we found when we used LPS blasts that cause murine B cells to proliferate, but does not cause human B cells to proliferate. And that was sort of a tip-off that LPS was not same thing as activating the B cells through their antigen receptor. We were also pleased to find that helper T cell lines were receptor positive because we had added our Tcgf, our human Tcgf, to murine helper T cell lines, and it worked fine. The fact that the scatter plots detected only a single line, straight line, led us to believe that we were only dealing with a single high affinity receptor site. There weren't multiple affinities in these experiments, but in terms of, as we'll find out later, as the story gets much more complicated, and there are other affinity sites on activated T cells. We'll get into that. The kinds of binding experiments that we did were very similar to the findings of EGF receptor binding, NGF, nerve growth factor, and insulin bonding. And those were the three peptide hormones that had been examined in this fashion at that time. We found that, as I demonstrated, that the binding was rapid and it was reversible, largely reversible. And so it would come to a steady state very rapidly within 10 or 15 minutes, 15, 20 minutes. It was also blocked by lysosome tropic agents. And we used ammonium chloride and also chloroquine of Trump's cure for COVID, because that inhibits lysosomes from functioning, and we could show that would block the degradation of the radiolabeled Tcga. So in all of these experiments, and at the end of our discussion, we said, well, this means that in immunology, we should be looking at endocrinological approaches in terms of how this system is regulated. So that was it. That was the end of the paper. And then subsequently, all kinds of things have happened in the past 30, 40 years. First of all, the paper itself became just a prototypic paradigm for all the rest of the interleukins that have been discovered. And we're up to like 43 or 44 at this stage of the game. And so everybody just looked at our paper and then repeated all these different kinds of experiments. And so that was very heartwarming. The binding assay itself was instrumental in what happened next in the IL-2 story and the IL-2 receptor story. It opened the way for the discovery of the structure of the Tcgf receptor, which became quite complicated. And now we know, 10 years later, between 1982 and 1992, that the Tcgf receptor is comprised of three different protein chains: alpha, beta, gamma. And without the Tcgf, radiolabeled Tcgf assay, we wouldn't have been able to discover those chains. The first chain, the alpha chain, was discovered just 1 year later, because a Japanese investigator, Takashi Uchiyama, when he did a postdoc with Tom Waldman at the NIH, he had immunized mice with the HUT102 cells. And he found that he could generate a monoclonal antibody that would react specifically with the HUT102 cells, but not other T cell lines. And when he looked at normal human lymphocytes, unstimulated cells, and that did not react, whereas antigen activated or PHA mitogen activated cells would react. And so Waldman's group contacted me because they figured that these are the characteristics of the Tcgf receptor, and we did collaborative experiments. And so in 1982, we published the first paper that showed that the composition of the Tcgf receptor, at least at that time, was comprised of a single chain that ultimately became called the alpha chain, and it had a molecular size on gels of 55,000 Daltons. So, and then we went on to it took us 5 more years to find the beta chain and 5 years more to find the gamma chain, and we'll get into that later. So the most important thing was this is all consistent with an endocrinological-like hormone that the immune system is using to communicate with them amongst themselves. And if you think about it, it almost has to be that way, because when life forms change from being unicellular bacteria and so forth and became multicellular organisms, there had to be a way that the cells in these multicellular organisms would communicate with one another. And so what we were discovering with T cell growth factor was basically that all of the cells in the body communicate via other molecules that function like hormones. And they have the characteristics of hormones. And those are, they function at very low concentrations because they have very high affinity receptors that are specific for each hormone molecule. They connote a very specific biological response on the part of the target cells, the cells that have these hormone receptors. And so I like to tell the story that when we got all these data and we were working on it and so forth and so on, I went up to Alan Monk's lab and I said, Alan, all these data mean that we're really dealing with a hormone, right? And he very, sagely and professorially said, well, I'm not sure you can call it a hormone yet, Kendall, because if it's a true hormone, there is always negative feedback regulation built into the system. Because otherwise, and in this particular instance, since the characteristic biological response was proliferation, if there wasn't some way that the hormone could turn itself off, you just have continuous and constant. Once it was activated, it would just keep on going, and that would be malignancy, which is also interesting. Is malignancy really due to the fact that the cells make and respond to its own hormone all the time? Well, we'll get into that later on in terms of negative feedback regulation, but that may take maybe another decade or two. I will stop there. Don't forget to like the video on YouTube, click on the bell, check out the website, the social media sites, particularly Twitter, and also Instagram. I have some paintings there that you might be interested in. So I'll stop there. Thanks so much. It was good.